Good morning, Sham Stars. In previous video of chapter 11 Biotechnology Principles and Processes, we learned separation and isolation of DNA fragments. In this presentation, we will talk about cloning vectors. Let's understand what are cloning vectors. Cloning vectors are the DNA molecules that can carry a foreign DNA segment into the host cell. It is a small piece of DNA taken from a virus, a plasmid, or the cell of a higher organism that can be stably maintained in an organism and into which a foreign DNA fragment can be inserted for cloning purposes. Most vectors are genetically engineered. The cloning vector is chosen according to the size and type of DNA to be cloned. Now let's understand features required for facilitate cloning into vector. Origin of replication, or we can call it ORI, selectable marker, cloning sites, vectors for cloning genes in plants and animals. We will discuss each feature one by one. Let's understand origin of replication, or ORI, is a sequence from where replication starts. Any piece of DNA, when linked to this sequence, can be made to replicate within the host cell. The sequence is also responsible for controlling the copy number of the linked DNA. Without ORI, DNA cannot be replicated in the cell. Now let's understand selectable marker. It helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Transformation is a process through which a piece of DNA is introduced in a host bacterium. The genes encoding resistance to antibiotics such as ampicillin and tetracycline are some useful selectively marker for Escherichia coli. Under the selective conditions only, cells that contain plasmids with selectable markers can survive. Cloning sites are required to link the alien DNA with the vector. The vector requires very few or single recognition sites for the commonly used restriction enzymes. The presence of more than one recognizing sites within the vector will generate several fragments leading to complication in gene cloning. Next feature is vectors for cloning genes in plants and animals. There are many which are used to clone genes in plants and animals. In plants, the tumor-inducing plasmid of Agrobacterium tumefaciens is used as a cloning vector. It delivers a piece of DNA known as tDNA in the Ti plasmid, which transforms normal plant cells into tumor cells to produce chemicals required by pathogens. Retrovirus, adenovirus are also now used as cloning vectors in animals because of their ability to transform normal cells into cancerous cells. So children, this is all about cloning vectors, plasmids, bacteriophages, cosmids, and features required for facilitate cloning into vector. I hope you must have understood. In the next presentation, we will talk about competent host organism. Thank you, Shem Stars. That's all for today. You can drop your queries on Google Classroom. Take care.